Welcome to Chapter 3. We get to look at biological macromolecules, things that we are very familiar with in most cases, at least when we're talking about food, and we're going to get into some detail on this. So let's take it from there. There are four major classes of biological macromolecules. So the term macro means large, and molecule obviously is molecule. So we're looking at compounds that are covalently bonded, and these are quite large structures as compared to some that we've seen. Now, the four main ones we'll look at are carbohydrates, lipids, which are aka fats, that's what we typically think of, proteins, and nucleic acids. Nucleic acids is the one group that I will kind of do a fairly brief discussion of because we get into that later on when we start looking at things related to DNA and genetics. All of them contain carbon, all of them. Carbon, hydrogen, and oxygen are the three main ones for carbohydrates. Lipids, same thing, carbon, hydrogen, oxygen. Proteins have carbon, hydrogen, oxygen, and the addition of nitrogen. So nitrogen's important here. We're going to see those amino groups, so functional groups show up here. And then nucleic acids have a couple of other ingredients that we'll see as well coming up. So macromolecules are made up of, so we said they're big, right? Macro meaning large, but they are, many of them have individual units, and these are called subunits, called monomers. So mono means one, and mer means unit. So a macromolecule is something that is built from these units, these monomer units. And when you get larger and larger macromolecules, they form something called a polymer, which means many units. All of the macromolecules join these monomer units by covalent bonds, but these covalent bonds have specific names depending on what we're looking at. So we're going to see carbohydrates, proteins, and nucleic acids are going to fit those descriptions. They're going to have individual monomer units, which then bond in particular ways to form larger polymer structures. Lipids is the one that is the exception, and lipids don't have a single type. So we're going to see that lipids can take different forms, so we can't call them having monomers. How they form, though, is the important thing. And in all cases, the way these monomer units come together is through this process known as dehydration synthesis. So you can see, so dehydration has to do with water, right? A loss of a water molecule, and then synthesis means to put together. So when water is removed from two monomer units, such as this down here, we're seeing this is called a monosaccharide. Here's another monosaccharide. See the arrow is pointing to what forms from those two, which is this disaccharide. Water is removed, and so our water molecule basically gets kicked out in order to form this bond. And here at the very end, if you could see it, it's kind of small, is the H2O. So that shows that where the water ends up is one of the products. So we have this disaccharide here, plus water, one molecule of water as a product. Okay. Hydrolysis is the opposite. So in hydro, which means water, and lysis, which means to break or to split, a hydrolysis is when polymers, which are large or larger, so here in this case we have these two, this is that disaccharide unit we just looked at, two sugars bonded together, they can also be split by the addition of water. So water can enter into these, where this bond is, and in doing so can break the bond and take the polymer, or this two unit thing, to single units, so back to their monomer states, where the H2O, you can see here's the OH here, so there's a hydroxide that entered onto this one, and then a hydrogen joining this one, again showing this, forming a hydroxide here. this is going to break this unit into these two individual units. So when we talked about, let me go back, dehydration, synthesis is removal of water, which causes the bonding of two of these monomer units. The bonds are covalent. And hydrolysis is the reverse. It's using that water breaks the bonds and then forms those monomer units again. So it's a reverse reaction. We're going to look at all of these reactions in terms of like how does the body do it? Does it happen quickly? Does it happen slowly? In all cases, there are going to the compounds that are going to be involved are called enzymes. And you've heard the term enzyme before. Now we're going to get more specific. So enzymes are biological molecules. In fact, they're actually proteins themselves that are going to help to either speed up a reaction, which is usually what they do, or at least control the rate of a reaction. And so they're going to speed up the hydrolysis and dehydration reactions. And in doing that, so these the dehydration reactions are going to form new bonds, right? Because dehydration reactions take monomers and form polymers. And so that's going to require energy. So energy is going to be part of the input to make these larger units. And then when hydrolysis reactions happen, the bonds are broken, and that's going to release energy. So we'll look at some of the specific enzymes involved depending on the group of macromolecules we're concerned with. Some of them, or at least a couple of them, you may have heard of. So in carbohydrates, it depends on which carbohydrate, but you know the term lactase, or at least you know lactose, which you've heard of as a sugar that is broken down by this enzyme, lactase. And then there's maltose, which is the sugar that's broken down by this enzyme maltase. Sucrase breaks down sucrose, and amylase breaks down a sugar called amylose. So all of those fit under this large umbrella of carbohydrates. Lipids or fats are broken down by lipases. And proteins, their names can be a little different. Here's one, an enzyme called peptidase, and here's one called pepsin. So those are enzymes that help to break down proteins. So the first unit you're going to be looking at in some detail is the carbohydrates. Yay, carbohydrates. We're going to look at them in terms of their chemical composition, and then we're going to look at the different types of carbohydrates that exist. But mostly we know some of this. So we know that carbohydrates are found in lots of things, lots of foods that we eat, particularly breads, grains. We find them in fruits, we find them in vegetables, and their main function for us is providing energy. And they do that by breaking down and forming ultimately some form of glucose that goes into our bloodstream and then gives energy to our cells. All carbohydrates are represented by this general formula. So if you look at the formula, you see there's a carbon, and then there's a water molecule. There's H2O. So this is the carbohydrate. This is a hydrated carbon. And usually there's more, so this N is saying that usually there's, certainly more than one carbon, but what you know is that there's always going to be this ratio. So for every carbon you have, you're going to have two hydrogens and one oxygen. So that's the ratio. The three main types we'll look at are the monosaccharides. So those would be the individual units, the monomer units of sugars. Disaccharides, so di meaning two, would be the units where two monosaccharides have joined by dehydration synthesis. And then polysaccharides, you have more than two, usually sometimes quite a bit more than two. So we're going to see these. There's so many ways to use the vocabulary in biology, and this, of course, is the challenge, because we just talked about sugars and carbohydrates almost synonymously. Now, and we've used the terms monosaccharides and disaccharides and polysaccharides. So now we're going to take it apart a little bit more. We're going to be more clear about each of these groups. So a monosaccharide is something that, remember, it's a single unit, usually has somewhere between 3 to 7 carbons, and it has this ending, just like what we learned just briefly before with hydrocarbons, but in this case we use the term OSE. So the suffix is OSE, and then it's always going to contain this functional group, this carbon with a double bonded oxygen. If this carbon with a double bonded oxygen is at the end of the molecule, then we call that something that's the aldehyde or aldose form. So the aldose sugars are we'll see these coming up. If this carbon double bonded oxygen or carbonyl group is embedded somewhere in the middle of that carbon chain, we call it a ketose. So all of these carbohydrates or monosaccharides can be kind of classified in one of these two. It's either going to be an aldose or a ketose. And then we can go further and say, all right, now that we know that, let's look at it in terms of the number of carbons. So if it has 3, we call it triose or triose. If it has 5, pentose. And so hexoses are 6 carbon monosaccharides. Here's some examples. I don't think this picture is all that good, at least from my poor eyes, but you can see that there are some that are listed above here. So there aren't too many monosaccharides that only have 3 carbons. So this would be a triose, 3 carbons. But this triose has the carbonyl group on the end, which makes it an aldehyde or an aldose sugar. And this one has it sort of embedded in the middle, which makes it a ketone type of sugar. So aldose, ketose. Both of them are trioses, 3 carbons. Both of them are monosaccharides. The ones below, you can see we're getting some other examples down here, and you can look and see that all of these have the carbonyl group, the one I'm kind of circling with my cursor, at the end of the molecule. So all of these are aldose forms, aldose sugars, because that's what makes these all aldose. But this is a triose. This is a pentose, there's 5 carbons. And this is a hexose, because there's 6. Here we also see that we can have isomers. So that term isomer, remember, means it's the same chemical formula, but just a different arrangement of the atoms. So all 3 of these have the same chemical formula, C6H12O6. But on the left we have glucose, middle is galactose, and the right is fructose. So see if you can see the differences between them by just staring at these molecules. Sometimes, and often actually, some of these linear structures form ring structures. So here you can see monosaccharides also can form these ring-shaped molecules, like we saw before with the aromatic structures. And they can actually go back and forth. They can mutate between being a linear structure to a ring structure and then back again. So, and it talks about this alpha or beta position. I'll get into this a little more detail. I need to do this in a better way than what we see it here. So we have these, here's 2 examples down here of a sugar. Here's one called a ribose, has 5 carbons. Notice that the top of that little pentagon has an oxygen on it. And then this one, which is fructose, has 6 carbons. The top of that little pentagon has the oxygen, and those other carbons, notice, are above the plane of this ring. So here's a ring and here's a ring, but you can see here's a carbon on this side, and there's a carbon over here, as well as the carbons that are on the edges of this sort of